Okay. All right. So, yeah, good afternoon, everyone. And first of all, I would like to thank all the organizers and the Sofium for this great opportunity. And it's my great honor to give a presentation in such a renowned symposium. So today I will talk about some of the lipid-mediated modulation. I'm working on the sensory biology, and so today's talk about the sensory ion channels regulated by the surrounding lipids. So as you may know, so this is a simple flow of the sensory processes. Any sort of the environmental stimulation or any environmental cues is captured by the specific type of sensory molecules in the membrane, typically the ion channels. And this consequently leads to the various type of the cellular or the physiological responses. And it's kind of, the central dogma for the sensory processes. And there are many the way of the modulation, but I'm particularly interested in the modulation of the sensory ion channels by the surrounding lipids. There are many examples has been already known. For example, like lipid components, the lipid raft, or the lipid signaling mediated by the PIP2, the hydrolysis, or its metabolized fatty acids, can directly or indirectly modulate the activity of the sensory ion channels. Another example is like a propagation of the physical chemical property of the membrane, just such as through the physical shape of the membrane, or the fluidity by the unsaturation level of the bilayer. Or the direct oscillation of the sensory molecule can determine the expression level or the localization of these sensory ion channels. So today I brought the two topics from our recent progress, and which is about the lipid-mediated modulation of the sensory ion channels. One is through the propagation of the membrane property, and the second topic is about through the gene expression, which is pretty new. Okay, let's get into the first topic. So this one, so I would like to start with the ether phospholipid. Probably, I guess many of you haven't heard about this lipid before, but this is actually one of the common lipid species in a plasma membrane. So you can see here, this is the structure of the typical the glycerol phospholipids. You know, this is the glycerol backbone, and the two acyl chains, and the phosphohead group. Now here's the structure of the ether phospholipids. Only the difference between these two, the typical glycerol phospholipid and EPL, is the binding between the first acyl chains in SN1 position. You can see it has the ether bond instead of the ester bond. So that's why these lipids are called ether phospholipid. But otherwise, all other structures are exactly the same, so this is only a difference. And these ether phospholipids is actually enriched, it's everywhere in our body, but particularly enriched in the specific tissues and organs, for example, brains and heart. Then we have the many ether phospholipid in these tissues. And the recent studies have identified the physiological importance of these lipids because these lipids are associated with aging and some neurodegenerative disease, such as the Alzheimer's disease or the Parkinson's disease. However, there's a physiological, the molecular function has not been identified so far. So, and I'm been working on the sensory biology using the fruit fly as a great model. And we attempt to identify any of the lipid regulatory genes enriched in the sensory neurons. So we dissociated the sensory neurons and performed transcriptome analysis. And we found that one of the candidate gene was the EPL, ether phospholipid related genes. So these genes are highly enriched in the many types of the sensory neurons in the fruit fly body. And particularly one of them called the AGPS. This is the bottleneck enzyme for the EPO synthesis. So therefore, we generated the AGPS knockout flies using the CRISPR-Cas9 technique. So here's the lipid profile of the CNS. And the wild type, so you can see the black bars, the typical profile of the glycosphospholipids. Lipids, and here's the ether lipids. So some sort of the ether lipids does exist in the fruit flies. But compared to this, the AGPS knockout flies, there's no obvious changes in the glycosphosfolipid profiles, but you can see all the ether lipids is gone, and these white bar is gone here. So this is AGPS knockout, is ether lipid depleting the flies. So this is the expression pattern of the AGPS in the fly larvae. And you can see this is the head part anterior, this is the tail part and posterior ends. You can see this is the bright spots in the near to the anterior, and this corresponds to the CNS. This is the brain and the ventral nerve cord corresponding to our spinal cord. And this is a subset of the neuron expresses AGPS in the central nervous system. And here, you can see the repetitive pattern on the surface of the fly body. These are the cluster of all kinds of the sensory neurons. This is a magnified picture, and including the polymodal, the sensory neurons. This accepting all kinds of the like environmental cues, like a temperature, mechanical stimulation, or chemicals. So this kind of corresponds to the C fibers in humans. So this data clearly indicates that the AGPS or the ether lipids are highly enriched in the sensory system, the nervous systems. Because we are interested in the sensory the functions, so we perform the thermotaxis assay using a very simple apparatus like this. So we created the linear temperature gradients on the aluminum surface by bridging this with the two separated aluminum blocks controlled by the water bath. And we release the larvae and let them freely move along for certain minutes under the dark conditions and check their preferable temperature. So here's the results. You can see this is a black traces control, and they show the peak distribution in around the 20, 23 degrees. And so this is within the so-called optimal range for them. So there's a very, very typical distribution of the wild type control larvae. Compared to this, the AGPS knockout flies, you can see they lost the peak distribution in this range, and they actually invaded into the very high temperature range, which is typically the noxious for them. So you can see the significant increase of the fraction of the larvae in the high temperature zone. So which means that the AGPS is required for the proper temperature sensation, particularly for the warm temperature range. So this is another type of the thermotaxis assay, the two-way choice assay. We set the two temperatures, 20 and 29 degrees. So we force them to choose one of these temperatures. Because the wild type larvae don't like the high temperature, like a 29, so they completely avoid and accumulating onto the left side, the cooler zone. However, the avoidance index was significantly reduced in the AGPS knockout larvae. And this was more obvious when we knocked down the AGPS in the TRPA1 specific neurons. So we knocked down the AGPS using the TRPA1 AV neurons. These neurons are located in the subset of the central nervous system, and which is already known to involved in the warm temperature sensation. So this is another type of the sensory function, so you can mechanosensation. So we poke the larvae with the tiny filament. So this is the kind of foam thread filament for the larvae, very, very tiny. When we poke the larvae, this is very noxious for them, and it shows a stereotypical the rolling behavior. This is perpendicular to the body axis, and they show this kind of a quick escaping behavior. So we measured this and found that at the specific strength, the AGPS knockout larvae shows a less avoidance to the stimulations. And the same idea, we knocked down the AGPS in a specific the sensory neurons, and here we found when we knock down the AGPS in the piezo positive neurons or TRPA1 CD positive neurons in the peripheral sensory neurons, then we saw the reduction in the response, like knockout here. So once again, AGPS should be important for modulate the mechanosensitivity at the individual level. So the next, process is because we now know the sensory molecules, we want to know this, how these ion channels activities are modulated. So here's S2R plus cells is a Drosophila culture cell line, which intrinsically do not have the ethyl lipid by themselves because they don't express the AGPS. But when we supplemented the 18HE, this is the precursor of the ethyl lipid, we successfully introduced the ethyl lipid in the membrane. So here we can see the nice, like 50% of the whole, the plasma membrane contains the ethyl lipid. So we use these two conditions to check the ion channel activity. So here's the TRPA1, the thermal sensor. We made the whole cell, and this is the batch clump, and we perfuse the solution to change the temperature, which is monitored by the probe nearby. So you can see both in the absence of the EPL or the presence of the EPL, we could see the nice activation of the TRPA1 by temperature, the heating here. In both cases, regardless of the EPL. However, if we calculated the thermal threshold for this activation, we did see the significant reduction of the temperature threshold for the activation in the presence of the EPL. So suggesting that the EPL is required to sensitize the TRPA1 channel to the temperature stimulation. So here's the same idea for the piezo channels. We express the piezo channels in SR plus cells and place a mechanical activation through the glass rod. And you can see in the absence of the EPL, we can see the very tiny activation, but this activation was significantly augmented in the presence of the EPL. This is the quantification. So here, once again, the EPL is very important for the sensitization of the piezo channels to the mechanical stimulation. And lastly, we check the membrane property. So thank you for the similar sense. She already explained a lot about the AFM. But here we scan the surface of the cell membrane with the AFM cantilever to measure the force, the feedback from the membrane. And the long story short, we found in the presence of the EPL, ethyl phospholipid, we found that the membrane got more stiff, simply to get harder. And in terms of the fluidity, we checked with the chemical probe in the lipid order, and in the presence of the EPL, we found the increase of the fluidity in the presence of the EPL. So let me wrap up this the first part. So TRPA1 is a thermal sensor, and the piezo is the mechanical sensors. And I found the integrative role of the specific lipid species called the EPL. The lipid is beneficial to finely tune the receptor sensitivity, probably through the propagation of the membrane property like the membrane tension and the fluidity, which is also important for the individual level. Let me go into the second topic. So previously, we found that the rhodopsin GQ-PLC to TRPA1 cascade is important for the thermotaxis of the larvae. I don't have time to talk a lot about this, but the young larvae prefers the warmer temperature, 24, whereas the old larvae prefer the cooler temperature. And in this case, they utilize the rhodopsin cascade for the thermosensation. And because this includes the PIP2 hydrolysis, we focus on one of the metabolites, the deoxyglycerol, DHE, and we seek the gene related to the metabolism or synthesis of this gene for the thermotaxis. So here that we found the interesting candidates, the three genes, CG1941, 42, 46, which may form the cluster on the genome with the conserved structure. And their putative function is MAG acetyltransferase, or MG80 in short, to generate the DHE using the fatty acid and the monoacetylglycerol, MAG, as a substrate. So we generated the true mutants of each gene using the CRISPR-Cas9 and tested the thermotaxis as I just explained. It's the gradient assay. So here you can see on the top two, the CG1942 and the 46, it shows the left shift of the thermotaxis is showing in the red or the blue traces compared to the black traces. And whereas the one of them that didn't show any changes compared to the control. So based on this, they seemingly escape from the high temperature. So we named these genes the BISH1. This is a Chinese, the word for the submarine, or the hisho in Japanese. So from here, I only focus on this one of these genes, BISH1, due to the time limitation. So here's another type of the thermotaxis assay, 2-HOS assay. In this case, we give them 20 or 24 degrees the selection. And the wild type prefers 24 degrees over 20. However, the BISH1 knockouts, they didn't discriminate these two temperatures, as you can see here. And we can rescue by the genomic rescue. So here, once again, the DAG synthesis gene is important for the discrimination of these, the fine temperature discrimination. And long story short, we found that one of the coolest temperature sensing neurons located in the tip of the anterior end of the larvae. These three neurons were important and expressed the BISH1 in these three pairs of neurons. So when we knock down the BISH1 only in these three pairs of neurons, recapitulate the no discrimination phenotype in a 2-HOS assay. And here we rescue the BISH1 knockout phenotype just by expressing the BISH1 only in these three pairs of neurons. We successfully rescue the thermal sensitivity. And these three pairs of neurons actually express as the ion channels called IR25A and IR21A. These are the structurally similar to the ionotropic glutamate receptors in humans, but they are actually the core temperature sensor and permeate the many ions like calcium. So when these neurons were stimulated with the cooling, and they show a nice activation like this. This is a two stimulation, activation. But if one of the receptors gone, the receptor neuronal excitability is almost diminished. So we check the thermal responsibility of these three pairs of neurons between the control and the BISH1 knockouts. Yeah, we show some movie here. So let's the heating, cooling activation, heating, cooling activation. Okay, one more time. If you compare these two, the cooling activation is much more in control compared to the BISH1. So this is a quantification. So once again, the cooling stimulation activates the three pairs of neurons nicely in control, but which was almost diminished in the BISH1 knockout. So this is not diminished, it's kind of half reduced. Yeah, so there were many questions and many possibilities, but we checked the expression level of the receptors, and we found that actually the two receptors, IR25A and 21A, were half reduced compared to the control level in the BISH1 knockouts. And here, in a knockout background, we could successfully rescue the mutant phenotype just by overexpressing the IR25A in these three pairs of neurons, or the IR21A. So which simply means that the expression level of these receptors are the keys for the thermal sensitivity. We further look for the mechanism for the expression regulation, and we found that the predictive binding site for the many transcription factors in these two neurons, for example, BISH1 and IR25A. And some common actually exists, such as the broad. And the broad is one of the transcription factors important for the development. And these genes or the specific isoform, Z1, Z2, Z3, Z4, all of them are half reduced in the BISH1 knockout, the background, compared to the control. And lastly, when we knocked down the broad, not BISH1, only in the three pairs of neurons, recapitulate the no discrimination phenotype in a 2-HOS assay, and the mutant phenotype, BISH1 knockout phenotype, was successfully rescued just by introducing the broadening transcription factor into these 3 pairs of neurons, as you can see here. Okay, this is the wrap up. So here we found that the BSHU1, which is the lipid metabolizing enzyme, somehow controls the expression level of the broad transcription factor, which is important for maintenance of the thermal receptors, the IR25A and 21A. And this is important for the animals to discriminate the slightly cool temperatures. So there are many questions remain. So the biggest question is how the BSHU1, the lipid metabolizing enzyme, regulates the expression level of the broad. So one of the possibility is that because the BSHU1 generates the DHE, and the DHE is a very famous activator for the protein kinase C, the protein kinase C might have some of the regulatory roles for the broad. Another possibility is because the DHE is a substrate for the lipid droplet forming, and the lipid droplet is a protective role against the oxidative stress. So there may be some connection between this stress and the broad expression. And the possibility is that the ecdysone hormone, this is a juvenile hormone, and is known to regulate the broad through the AMPK pathways. So there may be some connection between these lipid metabolism and this hormone. So this is still under the investigation. Okay, so these are the lab members, and I would like to thank the Dr. Sito for the first topic and the Shanmei Dan for the second topic. Okay, thank you very much for your attention.
Thank you very much, Dr. Sokabe. Paper is now open for discussion. Is IR25A and 21 complex? You mentioned that it's related to glutamate receptor. So, does it respond to any extracellular ligand such as glutamate?
Yeah, thank you very much. That's an important the question. Well, actually, they're structurally similar, but they lost the binding site for the glutamate. So they're not the glutamate receptor in case of the Drosophila. But the overall structure is quite similar. And then they form the heteromer. So IR25A and 21A form the heteromer to be a functional thermal receptors. So if the one of them is lost, so they lose the function.
Okay, and then a naive question is, are these complex IR25A and 21A, how is it thermosensitive? I'm a little bit confused by your data, but are these channels also sensitive to temperature?
Yeah, they must be the thermosensitive ion channels.
Okay. That's why you found a difference between the BSHU1 mutant and wild type.
Yeah, for the neuroactivity and the thermotaxis. Yes.
Any question? Okay. Please join me thanking Dr. Sokabe again for wonderful talk. We are going